Hi, my name is Emmet Francis, and I'm a fourth-year PhD student in biomedical engineering at UC Davis in the Heinrich Lab. I'm also a third-year ARCS scholar, and so I want to sincerely thank all the ARCS members for your continued support over these years. Now, in times like these, I probably don't have to work too hard to convince you that our immune systems are vitally important. In my work, I'm trying to understand the basic principles behind immune cell movement. Specifically, I'm asking questions about how forces exerted on or by these cells allow them to spread over a variety of surfaces, and how this relates back to chemical signals within the cell. In the body, immune cells regularly need to spread rapidly on surfaces. For instance, in order to respond to infection, they adhere to blood vessel walls and crawl out into the surrounding tissue. Then they navigate to the site of interest by following chemical trails. At the site of infection, specialized immune cells detect pathogens like bacteria and fungi, and they bind to them and spread around them, completely engulfing them. This process, known as phagocytosis, is the main type of immune cell spreading that I examine in my research. In my experiments, I mainly look at this process in the most abundant type of white blood cell, the neutrophil. I isolate these cells directly from human blood and examine their spreading dynamics in one of two setups. In the first, I use two opposing microscopic glass tubes, known as micropipettes, to hold a neutrophil and a pathogenic particle. Upon bringing the particle into contact with the cell, I get a close-up look at the process by which neutrophils engulf a pathogen. In another setup, I coat an entire surface with molecules to trick neutrophils into thinking that the surface is actually a pathogen. In this case, when I drop the cells onto the surface, they try to eat it. In this case, they can't complete the process, so they spread rapidly at first, and then they give up after a certain amount of spreading. Using this latter setup, our lab recently found that the speed at which neutrophils spread out on the surface doesn't change when we pack adhesion molecules more densely on the surface. Intuitively, we might expect that adding more molecules for the cells to stick to would allow them to spread faster, but this doesn't seem to be the case. Over the past year, I've worked extensively on computational models to explain this phenomenon. In brief, I tested two alternative models. In one, the driving force for neutrophil movement is adhesive attraction to the surface, while in the other, movement is driven by forces actively exerted by components inside the cell. Our experimental findings are only compatible with the latter model, in which active processes drive cell spreading. In my remaining PhD work, I plan to expand my work with these models, using them to explore not just cell mechanics, but also the chemical signaling events occurring within the cell. Thank you.